Hello dear students. So in last class we studied about introduction about the topic enzymes, where we studied how the enzymes are named. I gave you different terms for enzymes like holoenzyme, apoenzyme, coenzyme, prosthetic groups. Okay. Then we studied how the enzymes contribute in a chemical reaction. I told you that for any chemical reactions to take place, the transition state is required. And for that transition state, very high level of energy, that is activation energy, is required. And only when that high level of activation energy is attained by the molecules, reactants, then only they reach the transition state and the product is produced. In a normal reaction, in normal condition, very few molecules can attain that much energy and show the reaction. So what is the role of enzyme? We studied that enzymes actually lower down the activation energy. As a result, many molecules can reach that level of activation energy and they can participate in the reaction. As a result, the rate of reaction becomes very fast. So that was the action of enzyme. And we know that enzymes don't participate in the reaction. So after a product is formed, that enzyme can again be used for next reaction. Also, we studied about the mechanism of enzyme action. I told you about two concepts. One is lock and key mechanism, other is induced fit hypothesis. In lock and key mechanism, one lock, that is substrate, has got specific active sites having specific enzyme for it which acts like a key, and that key, when fits into that active site, it breaks the substrate and forms the product. Later on, I also explained you the induced fit hypothesis, which states that these active sites are not very specific. In fact, they are flexible and can show certain changes. As a result, these enzymes, they can work on different substrates depending upon the situation. So these active sites may be quite flexible. Okay. Today we will study the factors affecting the enzyme activity. There are many factors which affect the enzyme activity, but we will take up these four factors, which are temperature, pH, substrate concentration, and inhibitors. Let's study individually how these factors affect the rate of reaction. We'll begin with temperature. When you take temperature, it is seen that the activity of enzyme is in a very narrow range of temperature. And if you see through a graph, so with the increasing temperature, most of the enzyme work best at very narrow range of temperature. And that very narrow range of temperature is called as the optimum temperature. So the enzyme activity is best at the optimum temperature, given all other conditions are favorable. And the temperature below the optimum temperature, that is towards the lower temperature, the enzyme activity is very low, or you can say the enzyme is inactive. But this is reversible. Inactive but reversible, which means the activity is very slow when the temperature is low. But as you increase the temperature, the activity of the enzyme will increase and it will become optimum in the range. And with the lowering temperature, the enzyme activity goes down and a point comes where denaturation of enzyme takes place. Denaturation means the enzyme structure is destroyed, and this is non-reversible. When a temperature is reached where the enzyme has become denatured, because all enzymes are tertiary proteins and these proteins are folded three-dimensional structures, and when we heat it, the temperature becomes very high, the bonds are broken, and when bonds are broken, the protein gets denatured and the active sites are lost. And when the temperature is again cooled down, those fragile van der Waals forces, because of which there are so many bonds have been made, disulfide bonds have been made, so those bonds cannot be again formed in that same way. Therefore, active sites are totally destroyed. So, when a temperature is reached where denaturation takes place, then the enzyme activity is totally destroyed. So, this is what is the effect of temperature. So, optimum temperature is very narrow range, and this optimum temperature is generally between 15 to 20 degrees centigrade is the best. And as you increase the temperature, the activity will increase and then it will decrease. Okay, and below the temperature, the enzymes will act very slowly, and a point will come where there will be no activity, but that will be reversible. So this was the effect of temperature. So next factor is pH. When we consider pH, just like temperature, pH also has a very narrow range where there is optimum activity of the enzyme. Just like the temperature, it also shows a bell-shaped graph, which means that the enzyme activity is highest here, and this is called as optimum pH. Now, in case of pH, most of the enzymes, they work best at neutral pH. So generally, for most of the enzymes, the pH, optimum pH is when the pH is 7. But there are exceptions. For example, in case of pepsin, in case of the stomach, the environment is very active. So all the enzymes which work in the stomach, their optimum pH is acidic. So there the optimum pH may be 2 or 3, just like pepsin, rennin, okay. But there are certain enzymes like trypsin which act on intestine, and in the intestine the environment is alkaline or basic. So such enzymes, the optimum pH will be 8 or more. So most of the enzymes, they are showing optimum pH of neutral, that is 7, but exceptions are there. So their optimum is different than other enzymes. So it is quite specific with respect to the pH. So the bell will be same. For example, in case of pepsin, the optimum pH will be 2 or 3. If you decrease the pH like 1, it will be more active. But after some time, the activity will stop. And if you increase the pH, then its activity will be decreased. Okay, likewise. Same way, in case of, lipase, it acts mainly on the intestinal fluid. So its activity will be optimum at around 8 pH. But for most of the enzymes, the activity is a very narrow range. For example, the salivary amylase. The salivary amylase shows the maximum activity at 6.8 pH, which is neutral. Okay. So, and pH is quite specific with respect to the specific enzyme. So now we come to the next factor, that is substrate concentration. Let's see how the enzyme activity changes with the substrate concentration. Now the next factor which plays a very important role is the concentration of the substrate. Let's see how the substrate concentration controls the rate of reaction. For example, suppose when we increase the substrate initially, the enzyme action will increase. But a time will come when the enzyme action will be maximum. This is considered as Vmax. Vmax means the velocity of the reaction is maximum at this concentration. After that, even if you increase further concentration, the velocity of the enzyme action will not increase. So that is Vmax. It is because at that particular point, all the substrates, all the active sites of the enzyme will be occupied by the substrate, and there are no more active sites available for further substrate. So even if you increase the substrate concentration, then the enzyme activity will not increase. So that is called Vmax. Now, when we consider the concentration of substrate, we consider the concentration at which the velocity is half of the Vmax. So at here we will take the halfway. Halfway means the enzyme activity is half of the Vmax here. So we plot a graph here, and a point comes, a point where the concentration is half of the Vmax. So that concentration is called as Km value. Km stands for Michaelis constant. So for every enzyme, there is a specific concentration at which the enzyme activity is half of the Vmax. So that is a constant value. Okay. So when we work on the enzyme activity, we must know what is the Km value of a particular enzyme, and that is fixed because it's a constant phenomena, constant value. Okay. So this was the third factor, substrate concentration. Now we come to the next factor which affects the rate of reaction of the enzyme, which are called inhibitors. There are different types of inhibitors. Let's study about the inhibitors. So the inhibitors may be of different types. We can categorize them into three groups, which are competitive inhibitors, non-competitive inhibitors, and allosteric inhibitors. Let's see how are they different. Competitive inhibitors are those inhibitors which resemble to a substrate and they compete for the specific active sites. For example, there is an enzyme called succinate dehydrogenase. For this succinate dehydrogenase, the actual substrate on which it acts is succinic acid. But when instead of succinic acid, there is malonic acid, which has also entered the system. This malonic acid competes for the active sites because the active sites of malonic acid and that of the enzyme, they are similar to that of succinic acid. So this succinate dehydrogenase can act on both depending upon the concentration. So when in the system the malonic acid enters, it acts as an inhibitor because it will slow down the rate of reaction, because many active sites of enzymes will be occupied by this malonic acid. But this system is reversible because once the malonic acid is disintegrated or converted into product, then the enzyme is free for acting for next reaction. So the competitive inhibitors, they are competing for the active sites, and they are reversible. Okay. But when we come to non-competitive inhibitors, non-competitive inhibitors are non-specific. Here they are specific, but these non-competitive inhibitors are non-specific. They are commonly called as poisons, because they will disintegrate the active sites, and the enzyme cannot act further. It is destroyed completely. So the reaction is irreversible, non-reversible. So very good example are hydrogen cyanide. Hydrogen cyanide, when they enter any system, they destroy the active sites of the enzymes. As a result, the total machinery collapses. Okay? So these are non-competitive inhibitors, and they don't compete for any active sites. They are non-specific and irreversible. Now comes the allosteric. Allosteric inhibitors, they behave like competitive inhibitors, but here in allosteric inhibitors, the product of a reaction may act as an inhibitor. For example, suppose glucose is converted to glucose 6 phosphate by an enzyme hexokinase. This hexokinase, suppose this glucose 6 phosphate has accumulated in the system and it is further not being converted to next product. So this glucose 6 phosphate will act as an inhibitor, and it will bind to an allosteric site which is present in the enzyme for time being, unless the reaction proceeds. So commonly these allosteric inhibitors are also called as feedback inhibitors, which means when there is no further utilization of any product, the enzyme temporarily will stop that reaction, and whenever that product is further used up, then these enzymes, this product goes out of that enzyme, and enzyme is free to act again on the substrate. So this is also a temporary inhibitor and depends upon the enzyme rate of reaction of the product, whether it is being used further or not. Okay, so we have studied about these three types of inhibitors. So today we studied different factors which influence the rate of enzyme and their action. Okay, I hope these are very clear to you. In next lecture, we'll study about the enzyme classification. Thank you.